My name is Nokbonga Mbanzi-Porejo, also a PDP postdoc here at SAIAB. I'm excited to introduce my colleague, Kerry-Ann. Sorry, I just put on my video on. So Kerry-Ann is a PDP postdoctoral fellow here at SAIAB. Her research interest focuses on climate change, fish physiology, of organisms, as well as larval recruitment in coastal areas of South Africa. Kerry is not only dedicated to her research, but also values family. And I'd like to extend my congratulations to her and her partner as they embark on this new journey together as newlyweds. Today's talk should be focusing on investigating recruitment dynamics and rentation patterns of fish larvae in nearshore environment along the south coast, east coast coastline of South Africa. Please mute, accept our speaker, and you can ask questions at the end of the talk by typing the question Q&A session, or also you can just raise your hand at the end of the talk. Over to you.
Thank you so much, Nokobonga, for that lovely introduction and for your congratulations. Thank you. So yes, good morning, everyone. And sorry about my voice. I'm just recovering from a very nasty flu, so I hope everyone can hear me clearly. But today I have the privilege of presenting work that was led by Dr. Kieran Miller and myself. And I'm here on his behalf to share our findings with you, which I recently also presented at the 7th International Marine Conservation Congress. So the conference took place in Cape Town in South Africa at the International Convention Center recently now in mid-October. And I presented slap bang in the middle of the conference on a Wednesday afternoon, just after tea. So it was a little bit, but not from attending talks to networking and socializing at social events that was organized by the conference, as well as team excursions or team building excursions with the MEZ team to Oceans Aquarium and Cape Point as well. So a lot of fun was had, and of course a lot of dancing was done at the final function. Okay, all fun aside now, I'm going to go into the talk that I presented, starting off by introducing coastal habitats. So coastal habitats, including your nearshore zones, embayments, as well as estuaries, are crucial areas for early life stages of marine fish species. They provide food, they provide shelter, as well as favorable conditions for growth and survival. So understanding the critical role that nursery habitats play is key for marine conservation, as well as the management of fish populations. An early understanding of dispersal in marine fish suggests that movement of their early life stages, in larvae specifically, is mostly passive, so driven by ocean currents and hydrodynamic features. However, more contemporary research has revealed that fish larvae actually are more active in controlling their distribution. So as they develop and they start swimming, they rely on environmental cues like smell and sound to help them locate these important nursery habitats. And this active swimming, being horizontal as well as vertical, is essential for their recruitment, increasing population connectivity and basically supporting species replenishment. At the same time, you have adult spawning behavior taking place, which means that eggs and larvae can also be retained within nursery rehab. For example, if you have the adults spawning, it may help retain these local hydrodynamic features. It's shown that in these, like this spiral base shown in the image, macroalgae and other passive particles have shown to tend to accumulate there or collect there in these low energy areas like sheltered rocky headlands and being transported across shore from high energy areas like your exposed sandy beaches as seen in the image over here. So moving that way. But sampling in this high energy environment has proven very difficult. This in high energy environments, another high energy environment is like the southeast coastline of South Africa, which is also a very dynamic region with variable temperature currents and wind, which is also shown to have affected or affects larval distribution. And even though large-scale processes are well understood in this region, understanding how local or understanding the nearshore dynamics and their impacts and recruitment and retention remains less explored in this region. So it would be very beneficial to understand how these local hydrodynamics and biological physical factors may interact and shape recruitment and population replenishment. And this would be key in improving marine population management as well as protecting these critical nursery habitats. So the aim of this study was to look at or investigate the recruitment dynamics as well as the retention patterns of fish larvae in nearshore coastal habitats along the southeast coastline of South Africa. And in order for us to achieve this, we had three objectives. Firstly, we wanted to look at or compare fish larval assemblages across different habitat stations, being sandy beaches, which are high energy, mixed sand and rocky areas, and rocky reefs, which may be low energy areas, within 3 small bays. And then secondly, we wanted to assess how shallow coastal velocities and currents may be affecting these various habitat stations within the 3 small bays. And then thirdly, assessing the influence of local environmental factors on larval distribution mechanisms. So larval fish samples, they were collected from 3 bays along the southeast coastline of South Africa, namely Claremont, which represented in red over here, Kelly's Beach in Port Alfred, represented in blue, and Salt Bay in Port Alfred as well, represented in pink. And these bays are very similar to larger log spiral bays, which are often formed or which are formed by wave refraction around headlands. And then within each bay, we had 3 different habitats that we sampled from. And this included a rocky habitat on the west, indicated in purple, a mixed rock and sand in the middle, indicated in yellow, and sandy beaches on the east, indicated in green. So these habitat stations were basically chosen to represent a range of low energy rocky environments to a much higher energy sandy beach environment. So the larval fish, they were collected during spring low tides over a period of 3 months from October to December of 2022. And they were collected using a modified beach net, beach seine net that was operated by 2 people and pulled parallel to the shore in water depths of less than 1.5 meters for approximately a distance of 25 meters. And once we had 3 replicates were collected at each habitat station and for each embayment. And after each haul, the plankton samples were, well, the plankton was rinsed and sieved and preserved into bottles containing 99% ethanol. And then back at the lab, the samples were sorted and counted and identified to the lowest possible taxon, to using morphological features. And then we also did further identification using, or verification using DNA metabarcoding of 50 known samples of known species. And then on sampling days, we also collected temperature, pH, and oxygen at each habitat station and embayment. And we also collected water velocity using an acoustic Doppler velocimeter, again at each habitat station and embayment. But for the purpose of this presentation, I'm just going to be focusing on the water velocity results. So diving into the results, looking at the species composition, we collected a total of 17,356 fish larvae that belong to 22 different species, with the family Sparidae dominating, making up 98%. A popular Sparid Diplotis capensis, or commonly known as blacktail, was the most abundant species, making 87% of the catch, followed by our Sapa, or commonly known as Strepy, also a Sparid, and the Cape Stumpnose Raptosargus holubi, also a Sparid. The majority of the fish, excuse me, that were collected were in the post-flexion stage, so 83%, with very few in the earlier life stages being pre-flexion and flexion grouped together, making up 2%. And we had early juveniles of only 1%, with the remainder consisting of eggs being 14%. So looking at the total fish larvae catches by month and for each bay and habitat station, we find that there was no significant difference among months. And there were, sorry, my phone is ringing. Sorry about that. Okay, so I was saying that basically for the total fish larvae caught, there was no significant difference among months nor embayment. But we did find that there was a significant difference among habitat stations, and post-hoc tests revealed that the east, indicated in green, and the middle, indicated in yellow, was significantly different to the west stations, which had much higher abundances. And when the total catch was separated into developmental stage, the earlier stages being the group pre-flexion and flexion stages, again, similarly, we found that there was a significant difference in abundance according to habitat, but not according to month or bay. Again, with the western habitat station having the highest abundance. Then interestingly, when we did a permanova and to show any differences in community composition, we found that there were significant differences in community composition among habitat station and months, but not between bays. And when we further did a indicator species analyses, we revealed that there were significant associations for blacktail and your black-nosed clipfish at the west rocky habitat stations, and your Raptosargus holubi, your Cape Stumpnose, and your white stumpnose, Raptosargus globiceps, associating with the east sandy beaches. We found no significant indicator species for the middle stations. Okay, and then moving on to the water velocity measurements, we found that the east and middle stations had much stronger water velocities, as you can see, with the east being represented in green and the middle being represented in yellow. And we see that if you look at the velocity currents at Kelly's East over here and Claremont East, it's generally moving westward. And the west stations overall, we can see had much weaker current velocities. And when we looked at the total velocity magnitude, there was significant difference for both bay and habitat station, with Claremont East having the highest velocity of 0.97 and Claremont West having the lowest velocity of 0.19. So superficially, it would appear that local hydrodynamic features are influencing fish larval abundances, and our results show far higher densities in the low energy west habitat stations, where we also found high accumulations of floating debris. But when we consider the community analyses, we are finding significant differences in species inhabiting the different habitat stations. So this goes to show that while passive processes are taking place, active movement ultimately may be determining the final location of settlement. So in conclusion, the study highlights that both physical, being the currents, and biological, being the active movements of the fish larvae, may be shaping the recruitment fish patterns here in these embayments. And these findings are valuable for informing conservation strategies or efforts, especially during the early life stages where they are most vulnerable. And for us to better protect these fish species, it's essential for us to understand how these small-scale physical and biological factors may be interacting. And lastly, we need to conduct, or future research should then focus on figuring out what actually, like, the processes involved in this active habitat selection. So I just would like to thank you all for taking the time to listen. And I just want to acknowledge our funders and those of you who did help with this study, be it field, experimental support. It is greatly appreciated. And I welcome any questions. Thank you so much.
Thank you so much, Kerry, for your talk. Is there anyone with a question? Please, you can unmute yourself and ask a question.
Morning, everyone.
Morning.
Thanks for the talk, Kerry. I would like to know which guides do you guys use to tell the difference between fish larvae, since sometimes they may look alike.
Yeah, it's very difficult with fish larvae because obviously, you know, they still haven't developed the distinguished morphological identifications, but we use guides based, I think it's on the Australian fish guide.
Oh,
And I'm not too sure of the other one, but the person who was the pro behind that and identifying was Dr. Kieran Miller. I was mostly responsible for all the DNA things.
okay. Thanks, Kerry.
Thanks, Pumza.
Hi, Kerry. Lal here. Can you hear me?
Hello.
Yes, we can hear you.
Okay, thank you for the talk. I just have a quick question. For the breakdown in terms of life stages of fishes, do you find the earliest life stage is a specific species, or is it spread across, and how does that relate to the habitats that they're found in?
I think that we did find quite a lot of post-flexion in the west side, or the west low energy sides of the embayment. And I don't necessarily think it differed between the species, but I'm not entirely sure.
Okay,
Yeah.
no, thank you. I was just wondering, how that would correlate to where these
No,
adults are
thank you.
and how they're being transported from
Yeah.
however the currents are going to this West Rocky site. So if the earliest life stage is in the West Rocky site, I'm thinking that they're probably closer to where the adults are spawning, you know, and
Yeah.
closer to where, or they're moving there, and then once they at a stage where they can actually actively swim against the current, then they can move back into
Yes,
the more high energy environment. Okay.
I think so.
Okay.
Thanks, Lau.
Thanks, Kerry.
I think we still have time for some questions. If anyone has a question, I'm not seeing any in the chat box. No.
Hello, Kerry. Good morning.
Good morning.
It's Vota here. How are you?
Oh, well, and you, Vota?
Fine. Kerry, I was intrigued that you picked up some Clinid larvae.
Yes.
And lots of them or not too many?
There were quite a few, Vota. I think Kieran might be able to answer that a little bit better if he's online. But we did have quite a few clanids, and then again we had quite a bit of egg species, eggs that we still trying to identify using barcoding. So it's quite interesting.
Because it would be interesting to know how many species of Clinid you eventually land up with. You see, one of the puzzles we've got is that we actually have no idea how long that larval stage in Clinid is.
Oh, wow. Okay.
As you know, they are viviparous species.
Mm-hmm.
And the best guess from a colleague at NMU is maybe a week, but no more than that.
Okay. Interesting.
Yes.
I'll look into that, Vota, and we'll actually see the total number of clanids and where actually they're predominantly occurring in which habitat. And we can chat. Sounds very interesting.
Okay, will do.
Thank you, Vota.
Hi, Kerry. I think I have something on the chat. So QN said we only had one species of clinid, but they were very abundant, the third most abundant species.
So that answers your question, Vota.
That's actually quite surprising, since there are at least two other species which are very common in rock pools.
Okay. Cool.
Okay. Also, we collected a pre-flexion to post-flexion. That's a continuation of the answer.
Yes.
So what did I do? Any other question? I don't see, I think only have 2 minutes or sorry, I don't know what I did to my Zoom. Okay. No question? Okay, I guess if there's no question, then thank you so much, Kerry, for your talk. Yeah, I think
Thank you,
that will be the
everybody,
end
for
of the session.
attending.
Yes, thank you.
Okay. Thank you so much. Okay.
Thanks,
Bye.
Kerry.
Thanks. Bye. Bye,
Bye.
everyone.
Bye.